CRISPR stands for Clustered, Regularly Interspaced Short Palindromic Repeats. Now that's a mouthful that I'm sure you've already forgotten, but let's try to break that down. In 1987, a team of scientists first discovered CRISPR when they were studying the bacteria E. coli. What they found within the DNA of E. coli were groups of repeated segments of DNA. These segments were a mix between palindromic and non-palindromic. Palindromic means that they are read the same forwards and backwards, like taco cat or race car, but in the case of DNA, it would be something like ACT-GTCA. It wasn't until a few years later, in 1990, where Francisco Mojica put all the information into a nice and easy acronym. He saw the clusters of interspaced DNA that was short, about 20 to 40 letters, palindromic, and repeated. Francisco was analyzing another single-celled organism called archaea. When he found similar sequences to those published by the scientists from a few years prior, he knew that they served some important purpose. When analyzing the spaces in CRISPR, he discovered DNA that belonged to viruses, and Francisco hypothesized that the spaces coded an immune system for the bacteria. The findings would continue in 2005 when the protein Cas9 was discovered, and then in 2006 when a man named John van der Oost was the first person to directly program CRISPR by programming in the DNA for viruses before the bacteria even encountered them, similar to a vaccination for bacteria. The final piece in the CRISPR puzzle would come in 2012 when Jennifer Doudna and Emmanuelle Charpentier would discover how to hijack CRISPR and use it as a gene editing method. Using guide RNA and tracer RNA, they were not only able to cut out selected genes, but also replace them with completely new ones, taking CRISPR from a natural immune system for bacteria to a brand new form of gene editing. So now that you know a bit of the history about how CRISPR was discovered, let's try to break down how it works. So as I explained previously, CRISPR is a type of natural immune system for bacteria. So let's try to break this down. CRISPRs are the repeating segments of DNA that are shown here, and the other part of how CRISPR works as an immune system for bacteria is a part called CRISPR-associated proteins. Now basically, when a virus invades a bacteria, these CRISPR-associated proteins will come in and take a snippet of DNA from the virus. They will then implant that DNA into one of the spaces in between the segments of CRISPR. Now that DNA is in one of the spaces, a special protein called Cas9, so CRISPR-associated protein, will come in and take a copy of the DNA and turn that into RNA. So now that Cas protein has a copy of that virus's RNA, it will act kind of like a pair of scissors, and it will go out and search for the virus. Now when it identifies the virus with the same DNA, it will just chop it up, preventing the virus from copying itself and spreading. That's how the natural form of CRISPR works, but let's try to explain how CRISPR works as a gene editing tool. Gene editing CRISPR works quite similarly to the natural form of CRISPR with a few new added elements. You still have the Cas9 protein and the DNA, but now you have the new DNA that you want to replace. And then you also have guide RNA or gRNA. Now let's explain how these work. First off, you have the gRNA that is coded to the specific DNA that you want to replace or cut out, which would be this one right here. So first this will move in and go there. Then the Cas9 protein will cut out that DNA that you've selected, like so. And then finally, the new DNA will be inserted in place of the old DNA that you've just cut out using the Cas9 protein. That's a very basic explanation of how CRISPR works, but hopefully you get the idea of how the Cas9 protein works together with the guide RNA in order to replace selected genes with new genes. Gene editing has existed for a while, but what makes CRISPR so much better is that it is not only faster, cheaper, but it also is much easier than the alternatives. Now that you know how CRISPR works, here are some of the things that it can accomplish. In 2015, scientists proved the value of CRISPR by using it to cut out HIV cells in humans. Clinical trials for CRISPR-based cancer treatment started in 2016. Treatments for Huntington's disease, muscular dystrophy, cystic fibrosis, blindness, and many more genetic diseases are also possible. And imagine all the things you can do to a human. Scientists have already doubled the muscle mass in dogs, so just imagine stronger humans. When you make changes with CRISPR, that only applies to the person that is affected by CRISPR. The changes that CRISPR makes cannot be passed down to the offspring, which is where some controversy comes in. The current state of CRISPR is revolutionary, but we don't know what the future holds. When you make changes in the embryo, you are making changes in the person before they are born, but those changes can also affect their offspring, and the consequences decades down the line cannot be foreseen. Also, the unborn child cannot give consent to using CRISPR. This is why there was backlash against a Chinese scientist when he used CRISPR on two twins. He Zhankui was sentenced to 3 years in prison for conducting illegal medical practices in the embryos of two twins. The modification was to give the two girls immunity to HIV, and possibly have that immunity carry on to their offspring. Many in the scientific community stated their opinions on the matter, calling the treatment unethical, irresponsible, and premature. Doing this, he went against the bans made by the government, and he was sent to prison. Although he had good intentions, his treatment has many risks, many of which will not be seen until many years down the line. Although CRISPR has the potential to do many amazing things, it will take time before it is deemed safe to do so. Another controversy surrounding CRISPR is the concept of designer babies, not just making babies resistant to disease, but making them better. People are concerned that when CRISPR reaches its full potential, parents will start to design their own babies, choosing everything from hair and eye color to their muscle mass and even their intelligence, basically turning the process of making a baby into a video game character creator. Bigger muscles, resistant to temperature, heightened intelligence, turning the weak and pathetic humans into basically superheroes. With this technology, we have the power to play God. The only question is, should we? Thanks for watching my presentation on CRISPR. Hopefully you learned a thing or two about how CRISPR works and where this technology will be going in the future.